Why do we want blood flow? Because we want our good soldiers to come to that area where the insult has happened. So vasodilatation, vascular caliber increases so that more blood can flow. So there will be increase in blood flow to bring cells and proteins by increase in permeability. Now, this slide I will come to later. Now let's see how are these stimuli recognized by the host. This is the question we need to understand. How does the host understand that there is an insult going on? So this is very important. The recognition, this you will also learn in micro. How does a body cell, like suppose a neutrophil, if suppose we're talking about acute inflammation, how will the neutrophil know that microbe has entered into a body? For that, certain plasma membrane. So what is plasma membrane? It's the cell membrane. So certain cells have toll-like receptors on their plasma membrane. What are these receptors? These are receptors which sense microbes. तो माइक्रोब की बॉडी में कोई एक ऐसा पार्ट होता है जो ये प्लाज्मा मेम्ब्रेन के रिसेप्टर्स से सेंस हो जाता है. जैसे ये सेंस होता है, अ ट्रिगर ऑफ अ चेन ऑफ इवेंट स्टार्ट कि वो उसको बताता है कि चलो अब एक माइक्रोब आ गया है, अब हमारे न्यूट्रोफिल्स, ल्यूकोसाइट, मैक्रोफेजेस नीड टू कम इन. ठीक है. Then there are something called multiprotein inflammasome complex. Once the microbe is sensed, the microbe is taken inside the cell and is killed by phagocytosis. We will talk about this later. So how is the recognition done? The antigen presenting cells which are present in our body like lymphocytes or macrophages have toll-like receptors on their surface which recognize microbes. Now let's look into a normal vessel. Now we are talking about a vascular increase कि vessel की caliber बढ़ती है. ऊपर वाला जो diagram है वो दिखा रहा है कि ये एक arteriole है, forms multiple capillaries and post-capillary venule. This is a normal vessel which has normal same diameter. In the venule, there is no net flow of fluid inside or coming outside. Now, what happens during an insult? Once you see here is the tissue and there's an injury happening, the arteriole immediately gets dilated so that it allows more and more blood to come in. That blood comes in through these capillaries, and what happens in the venule? Because of some mediators, there is increased permeability. I'll show you how. Because of this increased permeability, the fluid from the blood column starts flowing into the telling you this that insulin is secreted causes vasodilatation of the arterioles which increase blood flow, and increase in blood flow gives you the first sign of inflammation which is redness. He asks why at a site of injury there is redness or heat. First, right, in to accumulation of okay. Hallmark of acute inflammation, which is increased permeability. Now let's look at vessel caliber in a three-dimensional structure of a blood vessel. You have the outer muscular layer and inner endothelium. Now this inner endothelium is composed of endothelial cells. Can you all see all the slides now are moving properly? Yes, ma'am. Okay, so these are the endothelial cells, single cells, which are actually part here. Is my arrow moving?
Yes, ma'am.
Okay, so please visualize this pink thing is basically this, which is the innermost endothelial lining. Now, what happens when there is insult, when there is vasodilatation? Once histamine is released, histamine tells the endothelial cells कि तुम contract कर जाओ. So, ये endothelial cells contract करते ही space create हो जाता है. This space is created, and this actually happens immediately. So that is why this is called the immediate transient response. Immediate but transient. मतलब होगा तुरंत. मान लो आपको suppose for example अगर आपको एक scratch हुआ, आए दिन हमें scratches होते हैं. स्क्रैच होने की इमीडिएट बात क्या होता है, वहां पे हल्की सी रेडनेस और स्वेलिंग हो जाती है, बट इफ यू हैव नोटिस्ड आफ्टर एन आवर वो चली जाती है। ऐसा नहीं है कि वो रेडनेस और स्वेलिंग पूरा 3-4 महीने तक रहती, नो। सो दैट इज व्हाई इट इज कॉल्ड इमीडिएट, क्योंकि वो तुरंत हुआ, बट ट्रांजिएंट, थोड़े देर के लिए रहा और अपने आप स्वेलिंग और रेडनेस खत्म हो गई। ये कौन करता है? हिस्टामिन, ब्रैडिकाइनिन एंड ल्यूकोट्राइन। इसके बारे में हम मीडिएटर्स में बात करेंगे। Now, contraction of endothelium also happens direct damage to the ileum. For example, burn हो गया, toxins are there in the body. किसी ने फूड, किसी को पॉइजनिंग हो गई, उसमें तब केसेस में क्या होता है? कॉन्ट्रैक्शन तब भी होता है एंडोथेलियम का। और कब होता है? जब ल्यूकोसाइट्स आके चिपक जाते हैं एंडोथेलियम में। न्यूट्रोफिल जब चिपक जाते हैं वो अपने टॉक्सिक ग्रैन्यूल्स रिलीज करते हैं और कॉन्ट्रैक्शन हो जाता है। लास्ट मैकेनिज्म of increased fluid response is transcytosis. What happens? This transcytosis mein there is no contraction of the endothelial cell, there is opening up of intracellular channels. So when there are see, you can see that there is no gap forming, but the channels here open up, because of which fluid comes into the extravascular space from the blood vessel. Now this chart summarizes the mechanism of extracellular fluid accumulation. कैसे होता है? या तो endothelial cell contract कर गया, डैमेज हो गया, न्यूट्रोफिल की वजह से डैमेज हो गया, या चैनल्स के थ्रू फ्लूइड बाहर आ गया। now so this what I summarized was all vascular events, ठीक है। now we will come down to the cellular events कि सेल्स के अंदर क्या चेंजेस होते हैं जब इंजरी होती है। so the general principle is this, that when there is an injury, there's influx of leukocytes to the site of injury. Which is the primary leukocyte which comes? It's neutrophil. These leukocytes, what they do is they ingest and destroy the injurious agent, be it microbe, toxin, necrotic tissue, by the process which is known as phagocytosis. We'll talk about phagocytosis in depth later. Now, the question is, how do leukocytes travel from vessel lumen to the site of injury? हमें पता है कि ल्यूकोसाइट्स ब्लड वेसल में घूम रहे हैं, न्यूट्रोफिल, मोनोसाइट, लिम्फोसाइट, ये सब ब्लड वेसल में। तो ये टिश्यू में कैसे पहुंचे? अगर आपको एक इंजरी टिश्यू में हुई है, वहां तक वो कैसे पहुंचते हैं? दिस इज अ वेरी कॉम्प्लेक्स प्रोसेस बच्चास, प्लीज अंडरस्टैंड। आई विल बी गोइंग इन वन शॉट, मेरे पास टाइम नहीं होगा रिपीट करने के लिए, सो प्लीज लुक इनटू इट very attentively. Now, how do leukocytes travel from the blood vessel? This is mediated by a multi-step process. It's not a single process कि भैया ल्यूकोसाइट आया और अपने आप निकल गया, नहीं, ऐसे नहीं होता है। ल्यूकोसाइट आया, आके वो अडहियर किया, उसको बुलाने के लिए कीमो अट्रैक्टेंट प्रोटीन्स निकाले गए एंड उसकी जर्नी शुरू हुई। Now look at this journey. This journey is divided into margination, rolling, adhesion, and transmigration. These are the 4 steps by which a neutrophil traverses from the vessel lumen into the tissue. Now, what happens? Look at this. When there is injury, we have talked about vasodilatation. Vasodilatation से क्या होता है? Blood slows down. When the blood slows down, the leukocytes, the neutrophils, which normally are placed in the center of the flowing blood में वो center में होते हैं, they start slowly moving towards the periphery. They gather along the endothelial cells, and this gathering is called margination. Now, this is an example of a normal blood flow. Normal blood flow में क्या होता है? Blood RBCs हैं ये, और ये purple colored ones are या neutrophils. They are all in the center of the lumen. जैसे ही stasis होती है या slowing होती है, कैलिबर बढ़ जाता है वेसल का, RBCs तो सेंटर में ही होते हैं, बट न्यूट्रोफिल्स क्या होता है? धीरे धीरे एंडोथेलियम के तरफ आके चिपकना शुरू हो जाते हैं। Now, इनको बुलाने के लिए we have a complete entire system which I'll talk later. This diagram shows that above is the blood vessel. You have a neutrophil. Stasis हो गया, ये आ गया एंडोथेलियम की तरफ चिपक गया, धीरे धीरे ये रोल कर रहा है। Rolling means coming from here and it is rolling and then it is crossing through to the tissue. So this is the site where the actual microbe is. So neutrophil has to come from here to here. It traverses this distance and then it comes like this. Now let's see this diagram in succession. In schematic presentation, please pay attention, see how it goes. See this. Could you see how the neutrophil rolled? What I just told you, this is how it is rolling. It has come, it is rolling, and it is adhering and coming down. Going back to this. Now, how this happens is in the endothelium. Now, the moment we want the neutrophil to come, the endothelium starts expressing certain molecules as selectin. Now, selectin is important for the margination part, where the neutrophils are coming and attaching to the endothelium. So there are three types of selectins, the ones which are expressed on the. So always remember this, that this is not never one side process. The expression of a protein न्यूट्रोफिल में भी होना होगा और एंडोथेलियम में. इट्स लाइक अ लॉक एंड की मैकेनिज्म. आप सिर्फ लॉक का कुछ नहीं कर सकते, सिर्फ की का कुछ नहीं कर सकते. द लॉक इज ऑन द न्यूट्रोफिल. नॉर्मली ये लॉक एंड की अपने आप सरफेस पे होते नहीं है, पर जहां इंजरी होती है ये एंडोथेलियम में एक्सप्रेस होना शुरू हो जाते हैं और न्यूट्रोफिल में. सो फॉर इन दिस एग्जांपल यू कैन सी दैट इन द एंडोथेलियम द पी सेलेक्टिन which is present on the platelets and E-selectin which is present on the endothelium start getting expressed, which has an affinity for the selectins which are present in the neutrophil. So these start recognizing so onto the endothelium and they start binding to it. Once the selectins and the neutrophil, they bind. On the neutrophil, the ligand modified glycoprotein. Now this, after this binding, what is important is सिर्फ बाइंडिंग से कुछ नहीं होगा, क्योंकि अगर आया तो ये फ्री होगा और ये खुल के निकल जाएगा. सो व्हाट इज इंपॉर्टेंट, adhesion is important, कि वो चिपक जाए इस पे. अभी सिर्फ इन्होंने एक दूसरे को एज अ लॉक इन की मैकेनिज्म आइडेंटिफाई किया है, पर इनका बाइंड फर्मली होना जरूरी है. उसके लिए हमें क्या चाहिए, उसके लिए हमें चाहिए इंटेग्रिन. Integrin क्या करेगा? Integrins are also expressed on endothelium like ICAM1 and which is expressed on the neutrophil like this integrin. So this integrin will bind onto this integrin causing a irreversible binding. अब once this form has taken shape, चाहे कितना ही high shear force blood आ जाए, this will not come out of its attachments. Till this stage, it can be uprooted, but at this stage, it cannot be. So this binds firmly to the endothelium by the help of integrins, selectins, and with the help of PCAM, it starts transmigrating. Look at this dumbbell-shaped neutrophil. Just to go through, हमने देखा था endothelium contract कर गया, separate हो गया. अब इस PCAM molecule से ये neutrophil अंदर आ गया. Now, what is important here is that this. Where is this PCAM present? This PCAM or CD31, like we are talking, is basically required for adhesion and it is present on the endothelial cell. Okay? Now this P-selectin, we need to understand that these molecules are normally, like I told you, are not expressed. एक normal cell में ये expressed नहीं रहती हैं, ये अंदर रहती हैं. Like this is a normal thing. ये अंदर रहती हैं. They are called Weber palette bodies. What are Weber palette bodies? They are basically intracellular store of in the endothelial granules of P-selectin. So P-selectin के granules हैं जो endothelial cell के अंदर है. जैसे ही histamine या thrombin आता है, ये ऊपर आके express करने लग जाते हैं. So these are Weber palette bodies. Now, after explaining this, now you can see it is marginating. अभी तक it is, see, even though if you see this step, यहाँ पे वो चिपका हुआ है, but blood आया, तो ये हट गया. Again ये roll कर गया. Again ये roll हुआ, और अब now the role of integrins have come in. So integrin के वजह से ये चिपक गया. You look at this. Integrin red ligand will bind to this blue ligand. There is irreversible binding. Now it has gotten fixed with the help of integrins, and with the help of PCAM, it will now transmigrate through to the area of injury to remove the bacteria. Am I clear?
Yes, ma'am. Yes, ma'am.
चलो, so we'll move on. Now, ये जो पूरा प्रोसेस हुआ, अभी हमने सिर्फ सेल की बात करी, बट ये प्रोसेस होने के लिए हमें मीडिएटर्स की जरूरत है. न्यूट्रोफिल को कोई तो बताएगा कि बेटा यहाँ आपको आना है, यहाँ इंजरी हुई है, वहां पे क्या आएगा? मीडिएटर आएगा. वो मीडिएटर की बात हम दूसरे लेक्चर में करेंगे, ठीक है? Now, so this is just telling you chemoattractants. Bacterial products are the chemoattractants. There are interleukins, cytokines, which we'll talk later. So you've understood chemotaxis. I am moving on the same diagram. Now, what is important is the type of infiltrate in a case of acute inflammation. What is the type of infiltrate in acute inflammation? It is predominantly neutrophils. When does it come? During the first 6 to 24 hours. So if somebody asks neutrophils कब तक चले जाते हैं, बिकॉज ये वाइवा में पूछा जाता है कि न्यूट्रोफिल्स कब तक निकलते हैं, कब तक आते हैं और कब तक जाते हैं, तो वो फर्स्ट 6 आवर्स में आ जाते हैं और 24 आवर्स तक चले जाते हैं. उसके बाद क्या आते हैं? मोनोसाइट्स आते हैं. Now, this is a small question which is again asked. Please mark it as a question. Why neutrophils only? ब्लड में तो बहुत कुछ घूम रहा है, लिम्फोसाइट, मोनोसाइट, इस्नोफिल, इतना कुछ घूम रहा है, क्यों? Why neutrophils predominate early in the acute reaction? Because they are more in number. They respond more rapidly to the inflammatory mediators than others. They attached more firmly to adhesion molecules, which I told you, क्योंकि इनके में integrins and selectins ज्यादा होते हैं. But they have a short life. That's I told you. 24 hours में ये चले जाते हैं. और ये कैसे जाते हैं? ये मर जाते हैं. They commit suicide. That kind of suicide is called something as beneficial suicide. उनके मरने से हमारे बॉडी में फायदा हो रहा है. That's why it is called a beneficial suicide. After they go, what happens? What comes? Monocytes. ठीक है? Now, ये तो हमारा general rule हो गया कि neutrophils come for acute inflammation. But there are certain infections जहाँ की neutrophils जाते नहीं हैं, वो रहते हैं persistent even after 24 hours. What are the examples? Pseudomonas infection. Neutrophils continue to be recruited for several days. Viral infections, lymphocytes. Viral infections में क्या होता है? Neutrophil नहीं होते हैं, lymphocytes ही आते हैं. And allergic reactions में क्या होता है? We know eosinophils predominate. Then what next? What happens after recruitment? एक बार recruit हो गए, they recognize the microbe or the foreign body, they activate the leukocyte and they kill by the mechanism of phagocytosis. Now, phagocytosis कैसे होता है? How do you recognize कि भैया इस सेल को हम फेगोसाइटोस करके खा के चबा के खत्म करेंगे या इसमें हम टॉक्सिन इंजेक्ट करेंगे? हाउ डू यू रिकॉग्नाइज? दिस रिकॉग्निशन इज डन बाय समथिंग कॉल्ड फेगोसिटिक रिसेप्टर्स. What are the phagocytic receptors? They are mannose receptors, scavenger receptors, and opsonin receptors. These receptors basically are present on phagocytes like macrophages, antigen-presenting cells. Because these receptors are present, microbes can be identified. They attach to the receptor and intracellular killing starts. So how? Phagocytosis involves 3 sequential steps. I told you one is recognition. So this on the right shows this blue thing shows a phagocyte. It has recognized this microbe. ठीक है? ये particles इस phagocytic receptor से attach हो गए हैं. This purple thing has come inside. A vacuole is formed, which is called a phagocytic vacuole. And inside the vacuole, there is release of enzymes leading to degradation of the ingested material. And this is how the killing of the microbe occurs. This is just history. I'm not going to go through. I'm cutting short. Now, the same thing जो मैंने यहाँ right पे बताया है, इसका एक schematic presentation है. ये देखो, ये phagocyte cell है, either neutrophil, monocyte, macrophage. उसके अंदर उसके inherent granules हैं. अब ये देखो, यहाँ पे red वाला क्या हुआ? Microbe को अंदर ले लिया गया. ठीक है? By forming pseudopod around it. ये हम पढ़ते थे क्लास 12 में, that a pseudopod is forming around the microbe and a vesicle is formed. This vesicle is important. It is called phagosome. ठीक है? This is important. This what is phagosome? It's basically a phagocytic vacuole around the microbe. Then there is fusion of the phagosome with the lysosome. Look at this. This is the fusion happening. Now lysosome, what is it doing? It's releasing its toxic granules into the microbe. And this entire composition is called phagolysosome. And what has finally happened? Those granules have killed the microbe and it has ingested it. ठीक हो गया? So, how has the killing occurred? Now this is important. Now what while we talk, we will talk about the pink granules here on. In pink granules में क्या है? Killing कैसे हुई? कैसे हुई? फेगोलाइसोसोम फॉर्मेशन हुई. अब वो killing कैसे होती है? ऑक्सीजन डिपेंडेंट मैकेनिज्म से. ठीक है? न्यूट्रोफिल और उसके अंदर क्या होता है? You have reactive oxygen species, reactive nitrogen species, and lysosomal enzymes. These reactive oxygen species, how is it produced? Again, short note. Look at this. So inside a neutrophil, there is something called a H2O2 MPO halide system. This halide system, now imagine, ये box को आप वो pink granule imagine करो, उस phagosome को imagine करो. Phagosome के अंदर क्या हो रहा है? माइक्रोब आया, उसके साथ क्या हुआ? रिएक्टिव ऑक्सीजन स्पीशीज निकल गए. कैसे? सेल के अंदर जो ऑक्सीजन है वो गेट्स कन्वर्टेड टू सुपर ऑक्साइड अनायन. हाउ? बाय हेल्प ऑफ NADPH ऑक्सीडेज. नाउ दिस सुपर ऑक्साइड अनायन gets converted to H2O2 in the presence of superoxide dismutase. The H2O2 hydrogen peroxide in the presence of myeloperoxide converts a chloride ion to a hypochlorite, and this hypochlorite is a potent antimicrobial. This antimicrobial helps in ingestion of the red, जो मैंने यहाँ दिखाया था, ये red granules is by basically using the hypochlorite mechanism. So this comes as a short note. Please elaborate the halide system present inside the neutrophil. And when you answer this, make this chart, make the neutrophil, give as many diagrams of the phagolysosome, fusion, killing, because this is what is the most potent bactericidal system. यही चीज आपकी रॉबिन्स का ये I have taken up a thing. Microbe is there. There is a phagosome fusing with the lysosome, granules coming in. Ultimate degradation, and in granules के अंदर क्या है? You have the reactive oxygen species, which is ultimately forming a hypochlorite and killing the microbe. ठीक है? Now this is the thing, same thing which is mentioned. Now then comes, after reactive oxygen species comes the other mechanism, which is the reactive nitrogen species. So nitric oxide, which is present inside the body in a harmless way, now gets started, is expressed in a way that helps us to remove the microbe in a similar manner like the reactive oxygen species. Nitric oxide in the presence of a superoxide anion gets converted to peroxynitrate, which is a highly microbicidal agent. ठीक है? Now nitric oxide जो है, it is expressed in three forms. How? Inducible nitric oxide, endothelial nitric oxide, and neuronal nitric oxide. This we will talk in the mediators. Now, other mechanisms of killing, I will just go quickly. Now, this is very important, but just neutrophil entrapment. This short note comes. What are NETs? Question comes, what are NETs? NETs are neutrophil extracellular trap. Trap means, हमें पता है English में trap means something wherein you are trying to trap somebody. So what are these? They're by definition, they are extracellular fibrillary network produced by neutrophils. ठीक है? What is it composed of? Basically a neutrophil. When we see this diagram, okay, this is a normal neutrophil. This is the neutrophilic nucleus and the granules. When we stain it fluorescently, we see that there's a pink granule and there's a green cytoplasm. During injury or process, what neutrophil does is it extrudes this nucleus, and whatever is left, the extracellular material along with fibrin forms a meshwork. That network traps the microbe and does not let it go out of that area, and it keeps it localized. So when you see this, ये पूरा जो है ये तुम्हारा एक्स्ट्रा सेलुलर ट्रैप बन गया है और न्यूट्रोफिल का न्यूक्लियस निकल के यहाँ आ गया है और यहाँ सिर्फ साइटोप्लाज्म रह गई है. ठीक है? So that is what an NET is. So it is composed of histone and associated DNA and antimicrobial peptides and enzymes. ठीक है? Now I'll quickly go through clinical examples of inflammation, acute inflammation. Like we have places like abscess is a classic example of acute inflammation. Where do abscesses occur? It can occur in any organ. Let's take an example of hepatic abscess. This is abscess feature showing increased neutrophils inside a blood vessel. इसकी आपको practical में slide दिखाई जाएगी. There is vascular congestion. Look at this RBCs. Blood vessel has increased. RBCs have come out. There's extravasation. Again, see on high power, it is showing 3-lobed structures. These are all neutrophils. of acute inflammation. ठीक है? All clear? Let's quickly, we have 20 minutes. We will just brush through the chemical mediators of inflammation. Now, with the basis of what I have taught, the basic mechanism you have understood of how inflammation happens, injury happens. Now we will understand the, can you see my slide of chemical mediators?
Yes,
ma'am.
Okay, so now we will quickly go through how these mediators act, because this also comes as short note. Okay? So mediators are divided into two types: the cell-derived mediators and the plasma-derived mediators. Cell-derived मतलब cell से जो mediators और signals आ रहे हैं और plasma से जो आ रहे हैं. तो cells, अब हमने पहली बात करी थी कि chemical mediators के हिसाब से we have three mechanisms of three phases. One is immediate early response within half an hour. Like I told you, scratch अगर होता है आपको, चोट लगती है, तुरंत आधे घंटे में एक रिस्पॉन्स आता है, which is the immediate early response. I told you it's because of histamine. Then is the immediate sustained response. Immediate है but थोड़ा लंबा है. ये कब होता है? डायरेक्ट डैमेज के लिए, जैसे आपको बर्न हो गया, कहीं गर्म तेल से लग गया या इस्त्री लग गई, इमीडिएट होगा, थोड़े देर तक रहेगा, वो तुरंत जाता नहीं है. The third is the delayed response, which is actually not understood well कि क्यों होता है, but it is important because delayed response में we can intervene therapeutically. Now look into the mediators. Please write down, question comes, write down the cell mediators and plasma derived mediators in acute inflammation. Role of cell derived ऐसा कुछ. So this chart is telling you what are the cell derived mediators and what are the plasma derived mediators. सेल डिराइव्ड मीडिएटर्स मतलब कौन से सेल से हमारे सिग्नल्स आ रहे हैं. मीडिएटर्स हम सिग्नल्स बोलेंगे. मास्ट सेल से आ सकते हैं, बेसोफिल से आ सकते हैं, प्लेटलेट से आ सकते हैं, न्यूट्रोफिल से आ सकते हैं. ठीक है. प्लाज्मा में कौन से मीडिएटर्स हैं जो कि हमारा पूरा कैस्केड ऑफ इन्फ्लेमेशन शुरू कर सकता है? हमारा क्लॉटिंग सिस्टम है, काइनिन सिस्टम है एंड कॉम्प्लीमेंट सिस्टम है. वी विल स्टार्ट विद द सेल डिराइव्ड मीडिएटर्स. Now, cell-derived mediators can be vasoactive amines. बच्चा, I am telling you, mind you again, ये पूरा जो सेकेंड लेक्चर में ले रही हूँ, ये वाला टॉपिक, इट्स ऑल मगिंग अप, सारा रट्टाफिकेशन है. इसीलिए मैं पूरा वर्ड टू वर्ड नहीं बोलूंगी, आपको दोबारा पढ़ना पड़ेगा. उसके बाद आपके पास कोई डाउट है आप मुझसे आके पूछें. और मैं अभी सिर्फ मोटा मोटा बता रही हूँ कि जब हमने वो बात करी थी कि जंग हो रही है, वॉर इज गोइंग ऑन, तो कैसे पता कि सोल्जर को कहाँ जाना है, कौन बताएगा? ये कौन बताएगा? ये चीज मैं आपको इलैबोरेट कर रही हूँ कि ये कौन बताएगा सोल्जर को कि कहाँ जाना है और जाके वहां पे क्या करना है. सो ये मीडिएटर्स बताएंगे. ठीक है. कौन से मीडिएटर्स हैं? आपके पास वेजोएक्टिव अमीन्स हैं, एराकिडोनिक एसिड मेटाबोलाइट्स हैं, लाइसोसोमल कंपोनेंट्स हैं, प्लेटलेट एक्टिवेटिंग फैक्टर्स है, साइटोकिन है, नाइट्रिक ऑक्साइड है. वी विल स्टार्ट विद वेजोएक्टिव अमीन्स. व्हाट आर द मोस्ट इंपॉर्टेंट कंपोनेंट्स? हिस्टामिन एंड सेरोटोनिन. They are among the first mediators to be released during inflammation, but ऐसे नहीं खुले घूम रहे होते हिस्टामिन. They are stored as preformed granules inside mast cells. This example, ये एक mast cell है, इसके अंदर ये छोटे छोटे granules हैं जो जब सिग्नल मिलता है अपने ये granules रिलीज करते हैं. They degranulate. ठीक है. कब होता है? जब फिजिकल इंजरी है, ट्रॉमा है, तुरंत ये जो रेड रिस्पॉन्स आता है स्क्रैच करने पे इज बिकॉज ऑफ वेजोडाइलेटेशन एंड हिस्टामिन रिलीज, जिसकी वजह से ये तुरंत रिस्पॉन्स आता है. ठीक है. Now, if histamine binds to the H1 receptors leading to vasodilatation, and it is the principal cause of immediate transient response of permeability. Next vasoactive amine is serotonin. Serotonin is equal to histamine, but it is not that potent. उसके effects इतने ज़्यादा नहीं हैं. करेगा वो भी vasodilatation और leukocyte migration. ठीक है. कहाँ पे होते हैं सेरोटोनिन? हमें पता है हिस्टामिन मास्ट सेल में होते हैं. सेरोटोनिन कहाँ होते हैं? प्लेटलेट सेल्स एंड न्यूरोएंडोक्राइन सेल्स ऑफ द GIT. ठीक है. Next comes arachidonic acid metabolites. Now, what are arachidonic acid metabolites? This is important. Short note on arachidonic acid metabolites comes. It's a basically polyunsaturated fatty acid. कहाँ से आता है? हम कहते हैं ना कि please eat healthy, eat PUFA, don't eat MUFA. Dietary sources से आता है. PUFA comes from dietary sources wherein it gets converted to essential fatty acid, which is linolenic acid. ठीक है? Now this PUFA is normally present in membrane, any cell membrane. But जैसे ही डैमेज होता है, डैमेज के साथ तुरंत phospholipase A2 enzyme release होता है that converts तुम्हारा membrane phospholipids into the active metabolites. This is a chart taken from Robbins. I will just summarize this in short. Cell membrane phospholipid होता है. जैसे ही injury हुई, क्या करता है? Phospholipase A2 gets expressed. It converts the membrane phospholipid into arachidonic acid. This arachidonic acid is further acted upon cyclooxygenase, lipoxygenase. That's why it's called a COX pathway. What is COX pathway? You will read this in pharma as well. COX pathway is this, that the arachidonic acid gets converted to PGG2, H2, I2, TXA2, D2 and E2. Beta ये सारा rectification है. The other pathway is acted upon by 5-lipoxygenase, which gives all these metabolites. Ultimately, what is happening is all these metabolites are causing vasodilation, inhibitor aggregation, vasoconstriction. ये आप लोग पढ़ लेना. This chart you have to mug it up. Then comes the third cell mediator, which is platelet activating factor. Now, what is platelet activating factor? As the name suggests, it's present in the platelets, basophils, mast cells, neutrophils. काम क्या है प्लेटलेट एक्टिवेटिंग फैक्टर का? बिल्कुल उल्टा. अभी तक हम क्या पढ़ रहे थे? वेजोडाइलेटेशन, कि भैया बढ़ेगा, स्टैसिस होगी. पर in a body you cannot keep allowing the same thing to happen again and again. ऐसे करोगे तो सारा there'll be a lot of loss of blood, there'll be a lot of loss of tissue, there'll be a lot of injury. So what happens is counter mechanism comes in, wherein it says ki stop it. How does it stop? By secreting PAF. What it does? Vasoconstriction. Okay? Now once vasoconstriction starts, neutrophils don't come, injury is curtailed. Platelet activating factor also increases neutrophil adhesion to endothelium, which we had seen rolling margination. Then comes nitric oxide. Nitric oxide के बारे में हम already पढ़ चुके हैं कि कैसे ये act करता है, कहाँ होता है. मैंने पहले बताया था, it is in the endothelial cells. It is a soluble gas that is produced by macrophage. What it does, how it works, it induces cyclic GMP, which initiates a series of intracellular events causing relaxation of vascular smooth vessels. So that means it will cause vasodilatation. Now, what is the half-life of nitric oxide? Only seconds. Thus, it acts only at a place which is right next to it, because within seconds it gets dissolved. तो वो दूर तो जा नहीं सकता, इसलिए पैराक्लीन मैनर में काम करता है. What are the three different types? I told you ENOS, INOS, and neuronal. Now, ENOS and neuronal, the endothelial and neuronal ones they normally are present at very low levels, but when they are activated, फटाक से वो increase हो जाते हैं. जो INOS है, वो induce होते हैं मैक्रोफाज के अंदर जब इंजरी होती है. ठीक है. अब इन्फ्लेमेशन में नाइट्रिक ऑक्साइड का क्या रोल है? उसके डुअल एक्शन है. एक कि वो वैस्कुलर स्मूथ मसल रिलैक्स करता है, तो क्या हो जाएगा? वेजोडाइलेटेशन हो जाएगा. और दूसरा, वो सेलुलर कंपोनेंट ऑफ इन्फ्लेमेटरी रिस्पांस को इनहिबिट करके रखेगा, कम करके रखेगा. ओवरगो ऐसा नहीं करेगा कि बहुत सारे न्यूट्रोफिल्स आ गए और बिल्कुल ही वहां पे गदर मच गई है और इतने एंजाइम्स रिलीज हो रहे हैं कि पूरा टिश्यू डिस्ट्रॉय हो गया. वी डोंट वांट दैट. We want the microbe to be destroyed. We don't want the surrounding tissue to be destroyed. And who stops that? Nitric oxide. Okay. It also reduces platelet aggregation, adhesion, and curtails the inflammation down. Now, this is a chart again taken from Robbins. When a question on nitric oxide and its function in inflammation comes, please make this chart of nitric oxide. You have the endothelial one. It gets released to the neutrophil, causing reduced leukocyte adhesion. That is why you can see the neutrophil is freely moving. It is not moving. It's not attached to the blood vessel wall. Now, inside the macrophage, I had taught you that INOS will secrete peroxynitrite, which is a potent antimicrobial and kill the microbes. Now comes important mediators. Now then comes the role of cytokines. Now, what are cytokines? Cytokines are basically cellular signals which are required for the neutrophils and macrophages to go to the principal source and start their action. So what are the principal cytokines we know? One is TNF, second is interleukin-1, and third is interleukin-6. So it's all because of these that you see the effects of inflammation. Like, for example, आपकी body में कहीं injury है, abscess हो गया, बॉडी को पता कैसे चलेगा? आपको कैसे पता चलेगा कि आपको डॉक्टर के पास जाना है, आपको कहीं प्रॉब्लम हो रही है, पेन इज कमिंग, हाउ? Because of secretion of interleukin-1 and interleukin-6. See, it says interleukin-1 gets secreted from macrophages and leads to fever. Fever होगा तभी आपको लगेगा कुछ है बॉडी में. We need to show ourselves. Interleukin-6 की वजह से आपके बाकी effects of pain will come. ठीक है? So these are basically, cytokines are basically mediators required to process the acute inflammation. ठीक है? Then comes chemokines. What are chemokines? They are important for recruitment of leukocytes to sites of inflammation and migration of tissues. Then comes cytokines in chronic inflammation. Today I won't teach about this because we will cover it up in chronic inflammation. Now, just in short, what is the principal action of TNF and interleukin-1? Once macrophages get activated, they secrete TNF or interleukin-1, which can have systemic effects and local effects. Systemic effects, I told you, fever हो सकता है, भूख नहीं लगेगी, नींद बहुत आएगी, थकान बहुत होगी. So अब अगर आपको लगता है कि आपको viral fever हो रहा है, बहुत थकान हो रही है, so you know why? Because interleukin-1 and TNF are getting secreted. Why? Because उस वायरस को निकालने के लिए मैक्रोफाज ये सब secrete कर रहा है. लोकल इफेक्ट क्या करेगा? वेजोडाइलेटेशन करेगा, इंक्रीज ल्यूकोसाइट एडेशन कराएगा, इन्फ्लेमेशन कराएगा. ठीक है? Then comes chemokines. Short note, what are the chemokines? Basically, it's a family of small proteins which act as chemoattractants. नाम से पता लगता है chemokines, chemoattractants. They are very specific for leukocytes. हर एक ल्यूकोसाइट का एक कीमोएट्रैक्टेंट है, हर एक ल्यूकोसाइट का एक कीमोकाइन है. कैसे? हम देखेंगे. ठीक है? There are 4 of them. Number 1, CXC कीमोकाइन. ये कीमोकाइन किसको बुलाता है? न्यूट्रोफिल को. अगर एक जगह पे CXC रिलीज हो रहा है तो न्यूट्रोफिल को पता है ऑटोमेटिकली मुझे वहां जाना है. ठीक है? So this chemokine, all the 4 chemokines are basically classified into 4 major groups depending on the arrangement of the cysteine residue. So in CXC chemokine, they have one residue separating each other. तो 2 chemokine के बीच में ये एक X residue. ठीक है? It's also known as alpha chemokine. What is the typical chemoattractant of this type? It is interleukin-8, not interleukin-1, interleukin-8, secreted by macrophages, and they help in calling neutrophils. This is the most important one. The next one is CC chemokine. The first 2 residues are conserved to each other. They are also called beta chemokines. ये वाले chemokines किनको बुलाते हैं? ये बुलाते हैं मोनोसाइट को, मोनोसाइट कीमोट्रैक्टेंट प्रोटीन, ईसनोफिल को, इयोटैक्सिन, लिम्फोसाइट को एंड मैक्रोफाज को. तो सेकंड कीमोकाइन, अगर आपको कोई पूछे कि एक जगह पे मोनोसाइट को आना है या ईसनोफिल को आना है तो कौन सा कीमोकाइन आएगा? इट विल बी बीटा कीमोकाइन और सीसी कीमोकाइन. Then comes third chemokine, which is only C chemokine, which has only one conserved cysteine residue. This is important for lymphocytes. And the final chemokine, which is a CX3C chemokine, it's called fractalkine, and इसका role बहुत ज़्यादा discovered नहीं है. So short note on chemokines, you have to mention all the chemokines and what do they do. Now, what are the functions? They stimulate leukocyte recruitment. They control normal margination, जैसे हमने बात की थी, rolling margination. ठीक है? Then comes the lysosomal granules. हम जानते हैं neutrophil, eosinophil, all of them have granules. What are the role? Now, neutrophils got 2 types of granules. This is a Viva question. What are the granules of neutrophils? You have primary granules and secondary granules. The primary ones are specific granules, secondary ones are non-specific granules. Details आप लोग को अपने आप you'll be taught in your small group learning. Then you have neutrophil elastases, monocyte, macrophages. Then comes neuropeptides. What are neuropeptides? They are secreted by nerves and some leukocytes. What is the important role? It is important in propagation of the inflammatory response. What is the classical of a neuropeptide? Is substance P. And substance P is secreted whenever there is pain. P for pain, that is why substance P. Substance P helps in transmission of pain signals, regulation of blood pressure, stimulation of secretion by endothelial cells, and vascular permeability. So all this was cell-derived mediators, mediators which came out from mast cells, macrophages, and all that. Then comes your plasma-derived mediators. Now, plasma-derived mediators are 4 important. the clotting system, the kinin system, the complement system, and the fibrinolytic system. So what happens in the kinin system? Kinin system, ultimately in physiology, we have read the clotting pathway, alternate pathway and classical pathway. So what we know is that factor 12 then converted to 12A, which ultimately converts kallikrein, prekallikrein to kallikrein, and this converts kininogen to bradykinin. And what are we interested in is bradykinin. This bradykinin acts as an early stage of inflammation. ये bradykinin क्या करता है? स्मूथ मसल कॉन्ट्रैक्शन करता है, वेसोडाइलेटेशन करता है, इंक्रीज वैस्कुलर परमेबिलिटी करता है और पेन भी करता है. तो अगर हमें पता है कि हमें एक जगह पे पेन हो रहा है, इंजरी हुई है, पेन हो रहा है, तो हमें पता है सब्सटेंस पी निकला है वहां पे और ब्रैडिकाइनिन आया है. ठीक है, दैट वाज द काइनिन पाथवे. देन वील टॉक अबाउट द कोएगुलेशन और द क्लॉटिंग पाथवे. क्लॉटिंग पाथवे में अल्टीमेट हमें पता है क्या बनता है, फिब्रिनोजन. एंड this fibrinogen, when acted upon by plasmin, converts into fibrinopeptides. ठीक है. Now, fibrinopeptides or fibrin split products. Now, what do this fibrin split products do? They increase vascular permeability and are chemotactic to leukocytes. Final is the complement system. This you will be also reading in micro. But I will tell you in brief what happens. What is complement system? Basically, they are a collection of soluble plasma proteins. These are plasma proteins which help destroy a microbial or offending agent. इस सिस्टम में 3 पाथवेज है. जैसे हमारे क्लॉटिंग पाथवेज है, ऑल्टरनेट और ये क्लासिक, इसमें भी होता है क्लासिक पाथवे, ऑल्टरनेट पाथवे, लेक्टिन पाथवे. ये तीनों पाथवेज ultimately whatever they do, all the 3 pathways converge to form a product called C3 convertase. Now look at this. You have the alternate pathway. This is a microbe. This chart basically is showing कि एक microbe 3 pathway से कैसे destroy होता है. चाहे वो microbe alternate pathway से आए, classical pathway से आए या lectin pathway से आए. तीनों pathways से ultimately क्या होता है इस microbe पे? C3A. These they converge and they form ultimately what is known as a C3 convertase. ठीक है, all the 3 pathways lead to the formation of an active enzyme called C3 convertase, which actually splits C3 to C3A and 3B. The 3B you can see will attach to the microbe. 3A, what will it do? It will call neutrophils for killing this microbe. ठीक है, so it is 3. Now this composite component of C3B will act on C5 to convert C5 to C5A and C5B, and ultimately the C5B will cause attraction of a membrane attack complex leading to lysis. So C5, ultimately what is important is C5A and C3A are effectors of inflammation. They will call leukocytes. ठीक है, I know I have rushed through this. This actually will be elaborated in micro and then you'll be able to correlate it better. Now this chart is a final chart which shows interrelationship between all the plasma derived mediators. We know the kinin system, the fibrinolytic system, the clotting system, and the complement system. So this is a chart which is showing how they interreact. So kinin system, bradykinin is formed. ठीक है, kallikrein will help in converging from plasminogen to plasmin. This plasmin will convert complement C3 to C3A. Kallikrein will convert C5 to C5A, leading to the membrane attack complex, which will kill the microbe. The clotting cascade, how will it help? It will form thrombin, which will convert fibrinogen to fibrin and will form fibrin split products, which we know increase vascular permeability. So these are the interrelationships between plasma-derived mediators. And these are the charts. I'm not going to tell because this is all rectification. कि fever में we can see interleukin 1 आता है, pain में prostaglandin, bradykinin आता है. So that's all for all the mediators. Mota, you have, I will just go through it, revise through it quickly. You have cell-derived mediators and plasma-derived mediators. When you have, we will just quickly go through the first chart we had. I have 2 minutes, 3 minutes left, so I'll just sum it up. This was the chart. Now, after reading all this, now you know there are cell-derived mediators and plasma-derived mediators. Cells are from platelets, mast cells, inflammatory cells, and plasma are the clotting, kinin, and complement system. Ultimately, whatever it is, all these get secreted whenever there is injury at a site to kill a microbe. these mediators are required. Why? So that the vascular changes accordingly happen and neutrophils and the fighting soldiers come in and they get the right signal to be able to kill the microbes. ठीक है? Is it all right? Could you guys all hear?